Bismillah ar-Rahman ar-Rahim. Our lecture today, inshallah, is about cholesterol biosynthesis and degradation. Starting with the definition of cholesterol, which is a soft, waxy, fat-like substance that's present in the bloodstream and all other tissues of the body. Cholesterol is 27 carbon compounds. This figure shows us the structure of cholesterol, composed of 4 fused rings. 3 of them are cyclohexane rings called ring A, B, C, and 1 cyclopentane ring called ring D. These rings together are called the steroid nucleus. This steroid nucleus is attached at carbon number 17 to a hydrocarbon chain with 8 carbon atoms called the hydrocarbon tail. At carbon number 3, we have a hydroxyl group, and we have a double bond between C5 and C6 in ring B. Because cholesterol have both polar and non-polar groups, we considered it as an amphipathic compound. في شغلة لازم نعرفها إنه any compound with a steroid nucleus is called a steroid. We call steroids with 8 to 10 carbon atoms in the hydrocarbon tail and the hydroxyl group at C3 in ring A sterols or steroid alcohol. ليش sterols or steroid alcohol? Because of the presence of hydroxyl group. Cholesterol is the major sterol in animal and human tissues, performing several functions such as being a structural component of cell membranes, maintaining its fluidity and permeability. Cholesterol is also a precursor for vitamin D and bile acid. It's also a precursor for steroid hormones such as estrogen and testosterone. So because of its biomedical importance, our cells need to assure a continuous supply of cholesterol. About 45% of cholesterol in the body is obtained from average diet, particularly food of animal origin such as meat, egg yolk, and liver. More than 50% of cholesterol is synthesized inside the body. Virtually all tissues with nucleated cells can produce cholesterol, but tissues of organs such as liver and intestine make the largest contribution to the body's cholesterol pool with 10% each. Adrenal cortex and reproductive tissues also have an important role. Synthesis occurs in the cytoplasm with enzymes in both the cytosol and endoplasmic reticulum. For our bodies, in order to produce cholesterol, we need acetyl-CoA molecules to provide carbon atoms. We need NADPH to provide the reducing equivalents and ATP to provide energy. Synthesis process starting from the production of a compound called 3-hydroxymethylglutaryl-CoA, HMG-CoA. Initially, 2 acetyl-CoA molecules condense to give acetoacetyl-CoA compound. This reaction is catalyzed by the cytosolic enzyme thiolase. Note that acetoacetyl-CoA have 4 carbon atoms, 2 atoms from each acetyl-CoA molecule. Then a third acetyl-CoA molecule is added to acetoacetyl-CoA to form HMG-CoA. This compound has 6 carbon atoms, and the reaction is catalyzed by the enzyme HMG-CoA synthase, also in the cytosol. Next step is the formation of mevalonic acid. HMG-CoA is reduced using NADPH releasing CoA to mevalonic acid using the enzyme HMG-CoA reductase. This is an irreversible step and is the rate-limiting step in cholesterol biosynthesis. Mevalonic acid is then phosphorylated using ATP to mevalonate 5-phosphate. Further phosphorylation of mevalonate 5-phosphate, also using ATP, will convert it to 5-pyrophosphomevalonate. Decarboxylation of 5-pyrophosphomevalonate using ATP form isopentyl pyrophosphate, which is a 5-carbon isoprene unit. IPP is then isomerized to dimethyl allyl pyrophosphate DPP using the enzyme isopentyl diphosphate isomerase. IPP and DPP then condense to give a 10-carbon geranyl pyrophosphate GPP. Another IPP molecule is then condensed with GPP, forming a 15-carbon farnesyl pyrophosphate. 2-farnesyl pyrophosphate combine, release pyrophosphate and reduce to give a 30-carbon squalene. Because it's formed from 2 FPPs and each FPP has 3 isoprenoid units, squalene is formed from 6 isoprenoid units. Each unit is derived from 1 mevalonic acid, and each mevalonic acid needs 3 ATP molecules to be formed. So, in order to produce 1 squalene, we need to have 18 ATP molecules. Squalene is converted to the sterol lanosterol by a sequence of reactions that use molecular oxygen and NADPH. The hydroxylation of squalene triggers the cyclization of the structure to lanosterol. The enzyme here is squalene monooxygenase. The formation of cholesterol from lanosterol takes place in the membranes of endoplasmic reticulum and involve changes in the steroid nucleus as well as the side chain. Methyl group at C14 of lanosterol is removed to convert lanosterol to 14-dismethyl lanosterol. Methyl groups at C4 are removed, so 14-dismethyl lanosterol is converted to zymosterol. Zymosterol is then converted to cholesterol by a series of reactions. Note the change in the double bond position to be finally in cholesterol compound between C5 and C6. Also note the reduction of the double bond of the side chain. Okay, so by now we are done with the steps of cholesterol biosynthesis. So how does our bodies control this process? Normally, we synthesize cholesterol at a rate of approximately 1 gram per day and consume approximately about 0.3 gram per day. How do we keep a constant level of cholesterol in the blood? As I said earlier, the rate-limiting step in the cholesterol biosynthesis is the conversion or reduction of HMG-CoA to mevalonic acid. So the enzyme used here, which is the HMG-CoA reductase, is the major control point of cholesterol biosynthesis. The first mechanism is called the sterol-dependent regulation of gene expression. Expression of HMG-CoA reductase gene is controlled by a transcription factor called the sterol regulatory element binding protein, SREPP. This transcription factor binds to a short DNA sequence called the sterol regulatory element SRE on the side 5 of the reductase gene. In its inactive state, SREPP is anchored to the endoplasmic reticulum as you can see in the figure. When cholesterol level falls, proteolytic cleavage occurs and releases the protein. The released protein is then traveled to the nucleus and binds to the SRE. Binding of SREPP to the SRE increase the expression of HMG-CoA reductase gene, so more HMG-CoA reductase enzyme produced. High levels of cholesterol blocks the proteolytic release of SREPP, and the SREPP in the nucleus is rapidly degraded. So again, and as a summary, low levels of cholesterol activates SREPP to produce more HMG-CoA reductase enzyme, so more production of cholesterol, while high levels of cholesterol inactivates SREPP, so less production of HMG-CoA reductase, less cholesterol biosynthesis. The second mechanism is called sterol-independent regulation of gene expression. We have two enzymes, the AMP activated protein kinase and phosphoprotein phosphatase, which control the activity of HMG-CoA reductase enzyme covalently. Dephosphorylation of HMG-CoA reductase enzyme activates it, while phosphorylation of it by protein kinase converts it to its inactive form. Concentration of AMP rises, then we are in a low energy state, which means that we don't need to synthesize cholesterol. This will inhibit the HMG-CoA reductase enzyme by activating the AMP activated protein kinase, which will phosphorylate HMG-CoA reductase, so inactivating it. The third mechanism of regulation is the hormonal regulation. The activity of HMG-CoA reductase is controlled by several hormones. Glucagon, for example, increases the activity of HMG-CoA reductase. It enhances the activity of AMP-activated protein kinase to phosphorylate the structure of the enzyme. Insulin, on the other hand, increases the HMG-CoA reductase activity. It prefers the dephosphorylated or active state of it. The fourth mechanism is the competitive inhibition. Remember that competitive inhibitors are compounds similar in structure to normal substrates. They bind to the enzyme at the active site and prevent the substrate from binding, so inhibiting its effect. And that's all about regulation of cholesterol biosynthesis. Now, how does our body eliminate this cholesterol? Cholesterol is converted to the bile acids cholic acid and chenodeoxycholic acid by the action of the enzyme cholesterol 7-alpha hydroxylase. Here, a hydroxyl group is added at C7 of the ring B in the steroid nucleus. Note the difference between cholic acid and chenodeoxycholic acid, which is the presence of OH group at C12 of the cholic acid but not in chenodeoxycholic acid. Both cholic acid and chenodeoxycholic acid are called primary bile acids. Conjugation of cholic acid with glycine by an amide bond between the carboxyl group of cholic acid and the amino group of glycine form the bile salt glycocholic acid. Conjugation of cholic acid to taurine give the bile salt taurocholic acid, while conjugation of chenodeoxycholic acid to glycine give the bile salt glycochenodeoxycholic acid, and conjugation of chenodeoxycholic acid to taurine give the bile salt taurochenodeoxycholic acid.